So this review is to help you study for the genetics test tomorrow. We are going to review a little bit of DNA. We're going to talk a little bit about Gregor Mendel, Punnett Squares, and all the things that you may need to know on your test. So first, let's talk about DNA. DNA is what's called a double helix. That is the shape that was discovered by James Watson and Francis Crick. So this is what DNA looks like, either of these. DNA is composed of a backbone that is made up of a 5-carbon sugar and phosphate, and it is made up of nitrogenous bases, specifically adenine, guanine, cytosine, and thymine. And so to match up with this, adenine matches with thymine, guanine matches with cytosine, cytosine matches with guanine, and likewise, thymine goes with adenine. Adenine and guanine, these are purines. Cytosine and thymine are pyrimidines. I guess the way I usually remember this is that American Girl seems, if you're familiar with that company, is kind of, dolls and all those different things. Kind of reminds me of innocence and purity, and so I always remember American Girl is pure. So that's how I remember that. As far as DNA replication, because last unit we studied mitosis and meiosis, which is a process by which cells make copies of themselves. And so in order to do that during interphase, they go through the S stage, which allows them to replicate or make copies of their genetic information. In order to do this, they have to copy all of their DNA. And so what we're looking at here is a strand of DNA. Now, there's a couple of enzymes that are going to help us with this. First step of replication is this enzyme right here. This enzyme is what we call helicase. Helicase is going to be the one that unzips the DNA. So it takes it, unwinds, and unzips it so that our next enzyme is going to be able to go through and read it. So what happens is we have our second enzyme, which is DNA polymerase. And this enzyme is going to go through and it's going to take a look at all of the bases that have been unlocked and unwound and unzipped, and it's going to pair the matching bases with them. So for instance, if our base here is T, it's going to read T, and it's going to match that with an A. If this one is a C, it'll match with a G. G goes with C, and A goes with T, and so on and so forth. So the polymerase will go along the side and make a copy. Now keep in mind that this is happening on both sides at the same time. Once this has all gone through, so helicase has gone through, divided the double helix, polymerase has gone through and made copies of both sides, then the backbones are going to reform. And you will have 2 brand new identical strands of DNA. This is the process of replication. So in review, number 1, helicase is going to unzip. 2, DNA polymerase is going to make a copy or pair the bases. And 3, we have the backbone reforming to form 2 new helixes. Other things to talk about. The man Gregor Mendel. Gregor Mendel was a monk who lived in Austria. And monks, they live in monasteries, and within these monasteries they have different roles that they need to play. Gregor Mendel chose gardening as his. And so with this came the introduction of the garden pea plant. And he started to see how the traits were passed down, and so using gardening and his other passion, statistics, which is a type of math, he was able to take and figure out the possible outcomes of the crosses that he made. Ideal things, he studied 7 different traits, things like height, things like flower color, pea color, and structure, texture. He studied all those things, seven different traits. And in doing so, he was able to come up with his 3 laws. Now, as far as things that really made this beneficial, pea plants do not have any intermediate traits. They only have one or the other, the dominant or the recessive. There's no mixing. We're going to talk about a few exceptions here in a few minutes to his laws. But his law of dominance really held true for the pea plant, and so it made it ideal. It also made it ideal because it only had a 90-day turnover, and so we had those 3 months, and he had his results, which was really, really nice. Terms to know. Dominant and recessive. Recessive is the trait that is hidden or can be hidden. Dominant is the trait that will be expressed if it is expressed. And so we have those different things. We also have genotype and phenotype and allele. And we talked about this a little bit today as well, but I'm going to just brush on it briefly. So we have an allele. An allele is a particular version of a gene, which we talked about today. So here is our chromosome, one from mom, one from dad. Here is the gene in question. We're going to say this particular gene, here's mom, dad, this particular gene is a capital letter B. So it's the dominant allele. This gene is the lowercase or the recessive allele. And so this is alleles. Now, two alleles, big B, little b, go together to form a genotype. Genotype is what your genes say about you, the information that is on them. In this case, this is a heterozygous genotype, meaning different. One is lowercase, one is uppercase. We could also have a big B, which would be a homozygous dominant, or you could have little b, which is homozygous recessive. Homozygous meaning the same. And so those are the genotypes, and they in turn then determine the phenotype. Phenotype is the physical trait, or the thing that is readily visible on the outside. It is your eye color, your hair color, maybe your skin tone, maybe your height, your ability to roll your tongue, hitchhiker's thumb. All those different things are phenotypes. Also, you can have disorders that are phenotypes: cystic fibrosis, hemophilia, sickle cell anemia. These are all different types of phenotypes or physical traits that go along with this. Now, I mentioned before that there are some exceptions to Mendels Laws. Mendel worked during the 1860s to come up with his laws, and they still are held true for the most part today. But again, Mendel was working with the garden pea plant. Garden pea plant and the human genome are a little bit different. And so we have some things that don't quite hold true. For instance, codominance. Codominance we talked about is kind of like blood type. Type A is positive, type B is positive, and then O is recessive. If a person has an allele for A blood and an allele for B blood, they are going to express both of them fully. So that's what codominance is, that both of them are expressed fully. There's also incomplete dominance. Incomplete dominance is usually like a heterozygous genotype. So something like black fur color as the dominant, and white is the recessive. And if you have the heterozygous genotype, you have gray. So incomplete dominance is that both of them are expressed, but just expressed a little bit. It's incomplete. It's not completely there. If you had something like this with fur color, like fur color black or white, like a pony, like a horse or a cow, you'd have both of them present for codominance. So you would have black and white spots or something like that. That would be codominant. So codominant, incomplete dominance. Polygenic. Polygenic is when you have multiple genes, poly meaning many, genic meaning gene, many genes that contribute to a trait, like eye color, hair color. That's why nobody, even though we have, two people may have brown hair, we don't have the exact shade or the exact same type of hair. Polygenic, the other thing with this is pleiotropy. So whereas this one, many genes contribute, this one gene contributes to many different traits. So it's kind of opposite of that. We also have some mutations, inversion, where a chunk is taken out, flipped, and put back in. Insertion, where you have something added. Deletion, where you have something taken out. And translocation, where you have something detached and added to a whole other chromosome. And so you have those 4 mutations to keep in mind. Not really exceptions to Mendels Law, that's the rest of these, but they are also good to know. One other thing that I'd like to talk about is something we call non-disjunction, which is a type of mutation as well. Non-disjunction means that the chromosomes during meiosis fail to separate. And so the types of non-disjunction would be monosomy or trisomy. And in this case, the person only has 1 copy. We should all have 2 copies of every chromosome that we have. 1 from mom, 1 from dad. In monosomy, we only have 1 copy in one of the chromosomes. In trisomy, you have an extra copy. So, last thing that I want to brush up on is the Punnett squares. I think you guys know how to combine Punnett squares, but the one that I'd really like to talk about a bit is the dihybrid cross. And so I will kind of address that. I'm going to just use a couple different random letters. I'm going to use B's, and I'm going to use D's. So those are our genotypes that we are crossing. So we'll make our Punnett square. And I'm just going to do the first one. I'm not going to do the whole thing. But you have to have all the possible gene combinations because that will be the possible gamete choices. So 4 eggs, 4 sperm possibilities. So big B, big D will be our first one. Big B, little d. Little b, big D. Little b, little d. This one will be big B, little d. Big B, little d. Little b, little d, little b, little d. So those are our possible combinations. Then when we combine, remember to put the B's together and the D's together. If you have the same letter, keep in mind that you want to put the capital letter first. If you have 2 different letters like this, like the B and the D, it's okay if the little b is first. It also helps, it won't get you so mixed up if you do this. So for like this one, for instance, you'd have big B, big D, little d. For this one, you'd have big B, big D, little d. Well, I hope this review session has helped. If you have any questions, feel free to send me a tweet at Mrs. Vandervorst, or you can shoot me an email. See you tomorrow for the test. Good luck studying.